তাহলে আচ্ছা শতরূপাদি, আমি বাংলাতেই তোমার ইন্ট্রোডাকশনটা দিই। হ্যাঁ, মানে সবাই এখানে আশা করছি সেইভাবেই দিই আর কি। আর দ্বিতীয়ত হলো যে
বলবো আমি ইংরেজিতে বলবো, তো মানে আমার বলাটা ইংরেজিতে হবে তো?
মানে যেটাতে খুশি, কারণ আমরা শুধু শুনে আনন্দ পেতে চাই। এবং তবে হ্যাঁ, ইংরেজিতে বললে যেটা হবে, কারণ কল্পেশ বোধহয় ওটাকে রেকর্ড করছে, তাহলে পরে যদি আমরা একটু ইউটিউবে দিই তাতে তোমার অসুবিধা নেই তো?
No, I don't have any problem.
Then it's very nice, very nice. তাহলে পরে আরো যদি সবাই শুনতে চায় আর কি, সবাই শুনতে পারবে আর কি, সেই কারণেই। আচ্ছা, তাহলে শুভ সন্ধ্যা জানাচ্ছি সবাইকে, যদি আমার এখানে এখনো সন্ধ্যা হয়নি আর কি, হবে হবে করছে। তা আমি মানে শতরূপাদির সঙ্গে সম্পর্ক বহুদিনের আর কি। হ্যাঁ, আমার মানে পাঠভবনের দিদি আর কি। তারপরে ইথোফিলিয়ার প্রত্যেকটি পদক্ষেপেই শতরূপাদির প্রচুর উৎসাহ দেখেছি আর কি। ফেসবুকে তো শতরূপাদি জানায় যে মানে আমরা এগোচ্ছি, ভালো করে এগোচ্ছি। আমরা খুব মানে সৌভাগ্য আমাদের যে শতরূপাদিকে পাশে পাচ্ছি এবং পাবো একদম যতদিন ইথোফিলিয়া থাকবে আর কি। আমি জানি যে শতরূপাদি খুবই ব্যস্ত, তবুও অনুরোধ করেছিলাম যে যদি একটু সময় বার করতে পারে তাহলে শতরূপাদির গবেষণা সম্পর্কে একটুখানি শুনবো আর কি। যে মানে আমরা আমাদেরকে তো একটু বেশি শুনতেই হয়, না হলে কি করে মানে একাডেমিক্যালি আপডেটেড থাকবো আর কি। তা আমি খুব খুশি শতরূপাদি আসতে পেরেছে। তা মানে সবার তরফ থেকে নমস্কার জানাচ্ছি। তুমি শুরু করো, আমরা সবাই অধীর আগ্রহে ছিলাম এই কদিন আর কি, শুনতে পাবো। হ্যাঁ, আর যেহেতু জার্মানিতে রয়েছে সেই জন্য আমি বলছি হার্লিটস ওয়েলকামেন। ঠিক জার্মানদের মতন করে বলতে পারলাম না, যাই হোক। হ্যাঁ, ওরকম চিৎকার করে করে বলে। হ্যাঁ, সেই জন্য আর কি। ওকে।
So I am very much thankful to Choyon and team and this group যে আমাকে ডাকার জন্য। So is my screen visible?
Yeah, sure.
Okay. So let me start, and thank you very much to Dr. Choyon and the entire team for inviting me today. And since most of the people here are students, so I will try to give you some idea that what cancer is, and I will try to give you like brief description what we are doing and what are the things going on nowadays in the world. Okay. So I will tell you the entire thing in a form of a story. So that's why my topic is the story of cancer. Okay. So to start with, first we should know that what is cancer. And basically, the cancer is a disease caused when the cells divide uncontrollably and spread into surrounding tissue. So why the name is cancer? So if we know, like, what is cancer? Cancer is basically the Greek name of crab, right? So if 1,000 of the crab bites you and you feel the pain, that kind of pain is felt during cancer. So that's why the disease got its name. And the disease is there from time immemorial, but we didn't know about the disease, like how it happens, how it spreads for a long time. So this is nothing but uncontrollable cell division or cell growth. And what happens is when there is something uncontrollable, we can't control that, obviously there should be some consequences. These consequences we see in the cancer. So how it is caused? It is caused by the changes to DNA, and most cancer causing DNA changes occurs in the section of the DNA called genes. These are the genes we know, everybody know about the gene, but still we should, like, for the definition we should learn. So therefore the genetic changes causes cancer. So you can ask me that only the genetic changes are important? No, there can be epigenetic changes as well. Means it will not pass on to the next generation. That will be a transient alteration, but that can also cause cancer. Okay. but mostly it is the genetic alteration. Now, the thing is, each person's cancer has a unique combination of genetic changes, and the specific genetic changes may make a person's cancer more or less likely to respond to different types of therapy. Okay, so the thing is, everybody is different, and there are different causes of cancer. It can be hereditary. There can be different viruses like HIV or HPV, which can cause, or hepatitis, which can cause us cancer. Then there is UV radiation, different types of carcinogenic chemicals, smoking. Okay, due to the environmental exposure, we can see there is like carcinogenesis happens. What is carcinogenesis? Carcinogenesis is the process of developing cancer. Okay, and this genetic changes can also happen because there are different types of error as the cells divide, but there are different mechanism as well, uh, by which at first the DNA tries to, like DNA damage repair systems are there. At first, that tries to repair it. Okay, so body is a machine, and obviously, when a machine is there, there should be some internal mechanism by which it tries to stop the progression. Okay, so there are different type of DNA damage repair systems or DDR. So what happens is the cancer causing genetic changes therefore accumulate slowly. Whenever there is a genetic alteration due to different reasons, the DNA damage repair system tries to stop it. But eventually, as the person ages, the DNA damage, like the DNA alterations get accumulated and leads to higher risk of cancer later in the life. So these are the major problems, and how people die. People die because of the cancer by a process called metastasis. Okay, what happens there? That the cancer cells break away from the original tumor, then it travels through the blood or lymph to a distant location of the body, and where they exit the vessels to form additional tumor. So there is a process called mostly epithelial to mesenchymal transition or EMT. So like 90% of the cancers are from epithelial origin. Okay, and so the epithelial cells is converted into metastatic into mesenchymal subtype. Okay, this is called epithelial to mesenchymal transition. Then it flows through the blood vessel, and then when they exit the blood vessel to some distant organ, okay, we it again converts back to the epithelial subtype, that is called mesenchymal to epithelial transition or MET. So this process is very important because when it goes to the other body organ, we call them metastasis. And suppose there is a tumor somewhere in the colon, you can see over here, right? And it goes to the lungs, we can tell it metastasized colon tumor in lungs. We don't tell it as lung cancer. So it is metastasized colon cancer in the lungs. So this is the simple process. And what happens, when there is abnormal cell division in some organ, so what does it do? It keeps on dividing, damage that particular organ, and people die due to that organ failure. Okay. So the like when you listen or when you know that a person is suffering from cancer and dying, so cancer doesn't kill the people, but the organ failures eventually kill the people. So, this is just an pictorial overview, like how the cancer spreads to the other parts of the body, and it can goes to any kind of organs. And this some type of cancer have specific inclination to go to a specific organ. This mechanism is called organ tropism. Okay. Why I am talking about organ tropism? Because we did some research on organ tropism. That's why I am telling a few words on it. Okay. Suppose you have triple negative breast cancer, like then there are higher chances to of the organ tropism in the bones, in the lungs. Okay. So that's the idea. So if we know or have some idea of cancer, we know that what are the different warning signs. So there can be coarse and dark, excessive hair growth, abnormal menstruation, back pain, belly bloat, okay, pelvic or abdominal pain, constipation, difficulty eating, nausea, vomiting, fatigue, increased flatulation, urinary symptom, painful sex, etc. So each of the cancer have their own warning. You have to identify and you have to consult the doctor as soon as possible, because the soon we can diagnose the disease, the soon we can combat it. Okay, so that is very important. So if we see the new cases worldwide in the both of the sex, we can see according to the Globocan 2020, for the female, breast cancer has the highest rate of um, incidence, okay, for the female, and for the male it is lung cancer, okay. And if we see the death also, like in which cancer there is a maximum death, we also see the same trend, that for the female it is the breast cancer, that is around 15.5%, and for the male it is lung cancer, that is 21.5%. So now we have to think about Indian scenario. In India, what is the most prevalent one? So in India, the statistics of 2012, it shows that um, the lip cancer, mostly like that is the oral squamous cell carcinoma or oral cavity cancer, pharynx cancer, is higher in both males and females than the US population. Okay. And another thing where India is basically, you can see a huge number of rise, that is cervical cancer. This two types of cancer, like all of them are, you can see like epithelial origin, they have epithelial origin, lip cancer, oral cancer, cervical cancer, they are having high prevalence in India. Okay. So from this notion or from this idea, I started my PhD on oral cancer. Okay. So and if we see that from 1990s in a lot of area there is a progress. Okay. In many measures we are trying to do, um, like we are trying to focus on the newer treatment options, we are trying to focus on the prevention approaches, we are trying to improve the quality of life or QOL of the patients, and we are trying to increasing the survivorship. Okay. These are the major aspects where we are working on nowadays. Okay. So understand that not only the things we are studying, this is, we have to combat the disease from multiple perspectives, right? We have to understand the disease, we have to understand the patient requirement, we have to understand how to prevent it, right? So a group of people should be there to take care of each area, right? So another thing is that, like a dress, you think about a dress doesn't fit everybody, right? So you have to go to tailor, or you have to tell your specification online about your dress material and you will wear it, right? In the same way, the previous concept of the treatment strategies, like one treatment fits for all, doesn't work at all for this disease. So the future medicine focuses on tailor-made medicine or personalized medicine, where for each patient, because each patient is different, each patient's cancer type is different. suppose two patients are there and they are having breast cancer, but their molecular subtypes are different, right? So for one patient, there should can be expression of one gene more, and for other patient, there can be another gene is taking care, right? So we have to consider that. You have to analyze that, and then we have to prescribe the therapy. So these are the things basically uh, we have to keep in mind while working, right? And with the implementation of the personalized cancer care, we can say there are lots of area, very interesting areas are there in the cancer biology, right? So suppose a patient is coming to the doctor, first thing is diagnosis, right? So in the diagnosis, you have to analyze that what are the molecular pathways is associated with the cancer. Suppose a patient is like suffering from the breast cancer, very good. We are going for biopsy. What is biopsy? Biopsy is just taking out the tissue from the patient. Then you like stain it, do histochemistry, and see it under the microscope, and then you just tell that whether the cells are normal or abnormal. Then you conclude that whether there is cancer or not. Fine. But what type of gene expressions are there? For that you have to do some certain immunohistochemistry or some specialized techniques you have to perform. But if you want to know that what kind of treatment has to be given, you have to do the gene expression analysis. So we have around 25,000 genes, right? And there are different ways of checking those genes together. And if we find that in that particular cancerous condition, few genes are overexpressed or underexpressed, we check that which molecular pathways are affected, right? And there are some specific genes which tell you like which are upregulated or downregulated or altered in specific cancer. We call them biomarker, right? So biomarker identification is very one of the very important area in cancer biology. Like from that you can understand that whether that particular gene has been altered or not, right? Then different types of instrumentation and informatics has been come, right? Why instrumentation? Like for the diagnosis, right? There should be a lot of biopsy is a painful process. When you are cutting a patient's tissue, it's a painful process. So if there is a, there can be any non-invasive treatment where without any blood test is the minimally invasive technique, but without any blood test, if you can tell, right, like using ultrasound, okay, then PET, positron emission tomography, CT, computed tomography, if you can tell that yes, there are chances of cancer, then you go for the second line of confirmation, like biopsy. Right? So that's why instrumentation is very much needed, and informatics. Why informatics is needed? Like if you have some patient data already in your hand, like what genes are upregulated in that particular cancer, you can have the idea what can be the probable biomarker. Okay. And on that biomarker, after checking the expression pattern of the biomarker, the doctors then can take the clinical decision and suggest what can be the possible treatment strategies. Okay. So there are different types of advances in the chemotherapeutic regions. So when people didn't know how to cure cancer, they used to burn that particular area. Okay. Eventually, new treatments have come. We came to know about different chemotherapeutic drugs. But the major problem with the chemotherapeutic drugs are that whenever you are trying to kill the cancer cells, eventually you are killing the normal cells as well. Okay. The the chemotherapeutic drugs can't differentiate what is good and what is bad. Okay. So like you can see over here, like in 2007, that chemo dosing half for low risk neuroblastoma is basically what people are trying to do to reduce the dose of the chemo to reduce the corresponding side effects. You might have seen that many people who undergo chemotherapy, they are losing their hair. What is that? That is nothing but the side effects of the chemotherapy. It is not that due to the cancer the patients are losing their hair. So again, the thing is the pediatric cancer where the patients can't tell anything, right? So there also people are trying to increase their long-term mortality like that. Then with the different, as I was telling a few minutes back, that there are different types of imaging advances we are trying to do, okay, where we can reduce the invasiveness of the cancer diagnosis. Okay, so we are trying to identify methods by which we can tell without doing biopsy whether the cancer is there or not, right? Even suppose there is a dysplasia. What is dysplasia? Dysplasia is a kind of cellular alteration you find in the pre-cancer stages. So suppose in the pre-cancer is there, you are seeing that there is dysplasia and you are trying to cut out the tissue. What can happen? It can aggravate it. Okay, the dysplastic cell can get converted into the cancerous cells because dysplastic cells can get back into the normal cells or it can get converted into the cancerous cells. Okay, but whenever you are just trying to cut them and see them, there are since there will be inflammation and inflammation is a like this is called hallmark of cancer. I will tell what hallmark is eventually. You are increasing the chances of carcinogenesis. So we are trying to get different techniques by which we can, like minimally invasive techniques, imaging techniques by which we can tell that whether that is a cancer or not. So then there is rise of immunotherapy as well, but that is a very costly procedure. So there is CAR T cell therapy, customized vaccines which are coming into the picture, and what happens here is the T cells are collected from the patient's blood, then they are reprogrammed and go grown in large number in the lab, and it is again sent back to the patient so that the T cells can um, act as a defense mechanism for the cancer cells. So what happens in the cancer patients? What happens in the cancer patient? They lost their they become immunocompromised, right? So to increase their immune susceptibility, this immunotherapy is suggested. And these types of drugs are recently being marketed, but they are very very costly. um, those are also coming into the picture. Then you might be knowing nowadays that a lot of vaccines are there, like a few viruses are there which can cause genetic alteration. Like human papilloma virus is the major cause of the cervical cancer, and they have two types of strains like HPV 16 and HPV 18. So, which can basically modify the host genome, and then they cause this alteration of the host genome. So basically you can have vaccine against those. You can have vaccine against hepatitis B, hepatitis C, and basically this hepatitis B, hepatitis C can lead to liver cancer as well. And even there are some cancers for breast cancers as well where you can have some vaccines as well. So these are the newer area where people are working on, like designing vaccines for different types of cancers as well. Then finally coming to the supportive and palliative care. So there has nothing to do with the biology. This is more like a supportive therapy. Like when you cannot treat any patient anymore, like the chemotherapy is not working, radiotherapy is not working, neoadjuvant therapy is not working. Then what you have to do is you have to like better the quality of life, and um, quality of life becomes the central focus of the oncologist. You have to reduce the pain of the patient because they become completely immobile and they can't do anything. So the family's support is needed. Okay, and there is the white blood cells counts also get decreased when the people undergo chemotherapy. So you have to support the person. So that is comes under the supportive and the palliative care. So new drugs are also being coming to the market, like filgrastim. So filgrastim. So this is used to treat the neutropenia that is caused by the cancer medicine. So in this area also people are working. So the area where I am working is the cancer genomics. So here you have to know that what are the different hallmarks of cancer. So in 2000, there are 6 cancer hallmarks were identified. And in 2022, a recent paper by Robert Weinberg and Hanahan, they are called the father of cancer biology. They have identified 14 cancer hallmarks. Okay. What is the meaning of the cancer hallmarks means? Like cancer is a disease where all the 14 things are going to be altered, right? So there is sustainable proliferative signaling means the first thing is evading growth suppressor. Whatever the genes associated with the growth, cellular growth, they are they stopped acting. They are called tumor suppressor genes. So they stopped acting. And the oncogenes, their expression, like which causes the cancer, their expression is upregulated. Okay. So first is evading growth suppressor means there is increase in the tumor suppressor genes. Avoid immune destruction, enabling replicative immortality because of the telomere. They get their replicative. They become immortal. The cells become immortal. And then there is tumor promoting inflammation. So there is activating invasion and metastasis. I just told what is metastasis is. And there is genome instability and mutation. Okay. Resisting cell death because of the oncogene, deregulating cellular metabolism. So a lot of things, 14 things, all of the things happen simultaneously. So you can understand how many genes are playing a role in the process. Okay. 14 process means how many genes. So what happened in 2005 is the Cancer Genome Atlas launches. So what is Cancer Genome Atlas? So more than 2000 of the patient genomes were sequenced. In 2000 we know that we got the first genome sequenced, right, for human. In 2005 around 2000 of the cancer genome patient of the cancer genomes were sequenced. And now what we do, we analyze those data. So basically we have a data, what is called, huge amount of data in your hand which is not analyzed. So we have the data in your hand and that is publicly accessible. We are trying to understand that data, that how, from what information we can decipher from this data deluge. Okay. So this database is also increasing. This is the work I do now. We have we have access of n number of databases. We analyze those data. We try to identify the biomarkers, so which can be targeted by different drugs. So this also again a part of our research where after identifying the genes, we're trying to find the targets, because when there is alteration in the gene, different types of molecular mechanisms gets altered. The different molecular pathways get activated, right? So we try to find out different types of drug targets which can bind with uh, the like we call them ligands. Ligand bind with the receptor, and we can stop the carcinogenesis process. Okay. So this genetic alteration basically causes uh, the variety in the genomic landscape of human cancers as well. And according to the expression of the genes and different types of cancer of our body, we can say there are more than 5,000 human cancers. Okay. So you can understand the complexity of the disease. Okay. And for that, there is a huge rise of the targeted therapy where we try to identify which gene is altered in that particular patient, and then we try to target that particular gene and then we try to solve the problem. And that's how the concept of the personalized medicine is coming into the picture, right? So first, in 1997, first targeted drug, rituximab came, and then eventually trastuzumab for HER2-positive breast cancer, imatinib, where um, came into the market one by one. And basically, these are gene-specific drugs. Okay, in the breast cancer, HER2 is one of the major um, gene which can cause breast cancer. So these are specifically different gene specific treatment. So what we can understand now, we have a lot of scientists, they are generating information, they are doing experiment, they are uploading their data. They are clinical researchers, we are doing the clinical perspective, they are working on the clinical information. Physicians are there, patients are there. So we are taking data from different levels, making a database. And now scientists are trying to understand the disease from the molecular disease modeling, using different types of data from different trials, treatment regimes, guidelines, literature, tests, etc. Okay. So, just I want to tell you a small story. I tell the story to my students as well because it's very interesting story. So, one physicist was there in the University of Southern California. So, basically, she was suffering of fourth stage of, I think, some cancer. Oh, I forgot the cancer, what type of cancer it is. So Shirley Pipke was ovarian cancer. So Shirley Pipke was diagnosed with the fourth stage of ovarian cancer, and she was a physicist and she was working in the TCGA. And doctor told that there is no treatment options left. What she did, she got her genome sequenced, okay, and then she matched with the TCGA, and then she found that what is the gene associated with her disease, and she suggested the doctors that. Please give me the treatment for this, because there were no options left then. It was 2016, and it was a miracle that that treatment worked, and she is still alive and she is still working. So this is the news, or this is an information, or this is a story where you can understand the role of data in our life. If we can use that data, what we can do. Okay. So we have a lot of biological resources where we can work. We can work on the biopharmaceutical development. So where we can work in the biologics repository, and see, we can work on the drug synthesis and the chemistry part. So since we know about what are the genes associated with the different types of cancer, now we can target them, and then like we can tell, yes, this ligand can work as a possible therapeutic strategy. So we have around, you can see, 2 lakh compounds. So it is not possible to check the 2 lakh compounds for each of the cancers, right? So there are different computational techniques by which you can do it. Okay. So this is called docking molecular, and with the molecular dynamic simulation, you can identify different types of drug which can work in cancer. So what you need for that, you don't need anything. You need knowledge. You need a laptop, and you can do by your own. Okay. So that is the idea. Then, in Shantiniketan, we have all studied in the environment, and we know that a lot of plants are there which have their own medicinal benefits, right? So the thing is, from there we have, from our childhood, we have learned that there are a lot of natural products we can use for these purposes. But now we have to channelize our knowledge towards better therapeutic strategy identification. Okay. So for doing this, n number of human tumor cell lines are there. Like cell lines are those things, I don't know. Like I think you should know. You know about the cell lines. So cell lines are basically an in vitro model to study the um, Like, we take out the cells from the body and then we culture it, immortalize it, and then we check the treatment of different types of drug. Then on the in vitro system, and then we check different types of molecular pathway in the cells, and there are huge number of the cell lines already available where we can use those cell lines. And there is huge uh, application of the information technology. We have so much of data, we have so much of knowledge, we have so much of information. So if we can apply those information technology for identification of the drug or the biomarkers, you can understand that what we can do. So here just I'm trying to talk about my research in a very um, like the way you can understand. So this, just a second. So this image basically uh, is called optical coherence tomography. This is similar to the image that we get in the ultrasound. So ultrasound gives you the image of the deep tissues, okay, but the optical coherence tomography gives you the information of the surface tissue. So if they have a probe and you can just touch it on your skin, and you can get this kind of information. So you will get the information about the epithelial layer or the subepithelial layer, etc. So what we tried, and in the right hand side, these are the histochemistry images or the biopsy images of different oral cancer and pre-cancer. So what we try to do, whatever the information we are getting in the biopsy. So biopsy is a painful procedure. You are calling the patient, you are cutting that particular area, then you are slicing the tissue and seeing it under the microscope after staining, right, with hematoxylin and the OC. So this is biopsy, right? So in the skin or in the oral mucosa, what are the different layers? The layers are epithelium, subepithelial region, etc. So what if we there is no need of doing biopsy? If we can get the information by some other non-invasive technique, then how can we do it? So what we tried to do this optical coherence tomography for this biopsy as a substitute for biopsy. And we found that with different image processing procedure, we can do that. We can classify the different types of oral cancer and pre-cancer. Okay. So this was very interesting research. And the left hand side, the same biopsy tissue, we have processed and performed Fourier transfer infrared spectroscopy that we do for chemical identification. So what I thought, or what we thought, like suppose we have 4 or 5 samples of each of the pre-cancer and the cancer. If we subject it to the FTIR, do we get any meaningful observation? What we found that after doing some statistical analysis, we can classify those samples in different subclass. So suppose we in a setup where there are no doctors, nothing is there, only FTIR machine and an person who know how to perform FTIR is there, and a tissue is there. People can go for FTIR and they can be sure that whether it is cancer or what type of pre-cancer it is, right? Because whenever you go to doctor as also, doctor, this biopsy is a qualitative procedure. Doctor tells by seeing the pathological report. This is a quantitative method. Okay. So this is how in this way you can quantify or surely tell in which class of your tissue falls in. And we also got some information from the spectral range that in the oral cancer, the protein, there is much more alteration in the protein portion, while in the submucous fibrosis, it is like in the more on the carbohydrate side. So those kind of information we could gather, and by multimodal methods, we could classify oral precancer and cancers. So that was my work that I did during my PhD. So during my postdoc, what I did is, like, as I told, like that was oral precancer, that is the highest cause of death in India. This is another disease where um, cervical cancer, right? So we have like I have talked about TCGA, and there are different other publicly available databases where you can get the microarray data or RNA sequencing data. So what you can do is you can get those data set, more than one, three data sets, and you can perform different types of statistical analysis to identify specific biomarkers. So you might ask me that why more than three data sets are needed? Because if we have more than three data sets of so there are higher chances that we are identifying the best possible biomarkers. Okay, and there are different types of other RNAs which can modify the expression of the genes. There are miRNA, that is microRNA, which are like non-coding 20 to 22 base pair RNAs, which can bind to the mRNA and alter its expression. And then there are long non-coding RNA, like which are more than 100 base pair long, and they can also bind to the mRNA and change the expression of the mRNA. So you can get different types of network of the RNA, and from there you can decipher meaningful information like which gene can be important in this process, or which pathway can be important in this process, or which mRNA, miRNA, link RNA interaction can be important in this process. Okay, or whether the biomarker you are identifying is giving you the idea, like whether it can be a survival marker. Survival markers gives you the idea that expression of that particular gene can tell you that how much survival option possibility is there if a person is suffering from cancer. Okay, so there are different types of statistical analysis we are doing by analyzing this data. Okay, and see, we are trying to use network analysis. So network analysis means we are trying to see the interaction between different types of mRNA, miRNA, link RNA. But you can see that the networks are very complex. The interactions are very complex. We cannot decipher, or we cannot get any idea from seeing this network itself. So these are called hairballs type of network, because you can see, if you see a hairball, you cannot, it is completely chaotic. You cannot decipher any information from it. So by different type of analysis using different tools, like here we have used cytoscape, you can see that we are identifying different interaction of mRNA, miRNA, long non-coding RNA, and their interaction. So this work was to identify that what, like uh, the androgen receptor associated triple negative breast cancer, if it is there, uh, then what miRNA can be useful. And we found that this MID27A3P is one of the very important one, and we want to validate that information via different experimental uh, by different experiments. So basically, we are getting the data. Like the way, by two way we can validate our information: via computational method in other data sets, or via experimental method. Whatever the systems we have in the cell lines, then we can perform the gene expression study using QRT-PCR, or via protein expression via using this Western blot. So these are the works basically we do. Okay, and that's is the story I wanted you to tell, that how, what we are doing, what can be done, and what is going on. So the November is the prostate cancer awareness month. So each of the month is basically important, have their own awareness strategies. Like October was for the breast cancer. So I work in the now in the area of breast cancer mostly, uh, but yeah, so I can take any kind of data. So now I am more focused on two ways areas. One area is the biomarker identification and understanding the role of different miRNAs, long non-coding RNAs, etc. In the another hand, we are trying to identify different drug targets which can like with the different types of molecular docking simulation, so that what can be the very good therapeutic targets. That are the things we are doing now. And I'm finishing here with the hope that cancer may have started the fight, but we will finish it. So thank you very much for listening. and happy to answer um, your questions.
Thank you, Satrupati, for the excellent talk. And
Thank you.
I think we have few questions actually because uh, the concept of cancer for me it's clearer now after the talk. So I have some basic questions. So first, I will ask one thing: that at what stage we are now? So are we improving in cancer research therapeutics, or we are in the same ground just before 5 years or 10 years?
See, the thing is, a lot of people are doing research in the cancer, and not only from the biological perspective. So few people are engaged in the on the therapeutics, few people on the diagnostics, few people are on like how to working on, they are working on quality of life improvement. So a lot of people are doing a lot of things. So all of the things are going simultaneously. It is not like that we are lagging behind in any of the case.
Okay. Another question is that do you think the governments, all of the governments globally, they are taking initiatives, and especially in India, the government is taking initiative to reduce the therapeutic cost, because you are telling that some of the drugs, they are very expensive, and it's very how to say,
There
not
are
very.
like ICMR have some special drive where they are funding for some specific type of cancer or some specific issues, but that is really nothing to like. It is a very, I will say, more research funding is needed, and more exposure is needed. Yeah, government is doing, ICMR is funding, but we think that much more funding is needed. So what we have seen
Yeah,
during COVID, like when COVID was there, then we got the funding for COVID. But if we had the funding before, we could do better, right? So it is like that.
yeah. Yeah, means no. What I was thinking, that when a economically weaker population, and they are having cancer, and when they are going for the treatment, it's a huge burden for them.
That's true.
Uh,
That no,
so the
actually, I have
pharmaceuticals.
not seen any. I have not seen that much information. I don't have much information. There are few government hospitals where the cost is quite low, like Adair Cancer Institute in Chennai. But the thing is, most of the time, what happens whenever a patient is diagnosed with cancer in private setup, the NGOs come out, and the NGOs try to help. Like you might have heard about Milap or some other keto, right? So they come out and try to do the fundraiser, but basically that is not going to help in long run, right? So
Yeah, exactly.
that's very costly. That's a very costly thing. And another few things are happening is like, you might have heard about Memorial Sloan Kettering Institute, which is a very well-known institute of US, and a few others like MD Anderson. They are trying to set up their own center in India, so that with the help of the telecommunication, the treatment can be accessed by the patient of like who might be poor, not poor, but
Mhm.
who can afford it. So that is the thing that is happening, and that is also there.
Yeah, exactly. I can understand. And thank you for this issue of this big data analytics and other things. So we were not aware of that. Okay, that you have so much of data set, and statisticians and computational biologists, they are constantly working on this medical data, clinical data. Rather, yeah, it's very nice. So hopefully we will be uh, overcoming this issue. I don't know, but yeah, we have to be positive at some point.
Absolutely.
I have two questions online. Actually, due to a technical problem, one of the members who he could not ask directly, so he just requested me to ask on behalf of him. So, Shouresh, I'm asking your question. So, one question: this is chemotherapy has also genetically altering. components. Thus, is using chemotherapy does backfire in the way to revive cancer? Do you think?
This is a very nice question, actually. That is what I was trying to tell when I was talking. That the chemotherapy, what happens during chemotherapy, it doesn't differentiate between the normal cells and the cancerous cells. So it is given in the cycle. What happens is when you are giving chemotherapy, the cancer cells are dying, and the normal cells where the genetic alteration is happening, there a window is given, suppose for 7 days to 10 days, where the DNA damage repair tries to repair the normal cells.
Okay.
Okay.
Yeah.
Then again the next cycle is started when again the chemotherapy tries. But even when, like, if the DNA damage repair system doesn't work, obviously, so the person is already suffering from cancer, so it might give you a new mutation, and might be cancer stem cell can be created when the chemotherapy is given. And then what happens? The resistance, the problem of chemo resistance comes. So
Yeah.
it is not a kind of backfire, but this is the constraints of the chemotherapy, you can tell.
Okay. Okay. Thank you. Another question from him that what is the fate of nanoparticles, specifically plant-derived nanoparticles in cancer treatment?
This is also a very good question, and the people are working in the area. So I was talking about targeted therapy, right? So the targeted therapy is like where you can target directly the cancer cells where the normal cells will not get affected. Okay. So suppose if you are using iron nanoparticle or ferric nanoparticle, so what happens if you are just injecting the nanoparticles in the cancerous area, and just using the magnetic property to just where the cells, like the nanoparticles are accumulated, those cells will only be killed. Okay, so this is a part of targeted therapy, as I was telling. So like nanoparticle based treatment is already coming into the picture. And now coming to the plant, the second question, that is the plant portion, right? So there are already few chemotherapeutic drugs which we get from the plant. You might have heard about Vinca rosea, which we call Noyantara in Bengali, right? So
No,
the
yeah.
Yes. So vincristine and vinblastine is basically we get derived from these plants, and they are already well-known chemotherapeutic agents. So suppose if a drug is not cannot be delivered via systemic route, then we can make nanoparticles with those plant, and then we can use those. But I don't think plant-derived nanoparticle has any difference in the nanoparticle that are grown in different other ways.
Yeah, exactly. Thank you. Thank you. I have another question online. Actually, he also had some having some problem technical. Does epigenetics play a role in cancer? She's asking. She's Tithi Paul. She's a new member of Ethophilia, and she's just BSc second year now. So she's asking this
This is
question.
a very nice question. I was telling, right, that there are, yes, epigenetic changes causes cancer, and there are the DNA methylation is a very important plays a very important role. So the only thing is, epigenetic reprogramming is possible to cure the cancer as well. DNA methylation is there, then adenylation is there. A lot of epigenetic alteration also causes cancer. And you know, DNA like methylation based, like sulfite uh, associated, like microarrays also we do to get the idea whether epigenetic alteration is causing the cancer or not.
Oh, okay, okay. Thank you, thank you. So I have another question from someone having the same problem. Srinonti, she's asking, um, is there any biomarkers so that we can understand at which stage the cancer patient is? This is the first question.
Yes, yes, absolutely. The work I was showing for the cervical cancer, that was, can I just share my screen again? Or can I
Yeah,
get,
sure, sure.
okay.
Kalpesh, or
Yes,
Meghna,
yes, no
anyone?
problem.
Uh,
Yes, yes, no problem.
Sure. So this was the work we did is basically to identify stage-based cervical cancer and biomarkers. So basically, we identify specific markers which you can tell about at which stage it is. See, so the thing is, stage we define according to the a number of lymph node affected or whether the metastasis has happened or not. LNM classification it is told, like how many lymph node have been affected and then metastasis have been happened or not. So this is a completely a qualitative aspect. So what we tried to do, I tried to identify that what are the different biomarkers which will be able to tell that in which stage the cancer is. So this was our work only. You can just check it in the Google Scholar, which was published in 2019 in Gene. But
Very
yes,
nice.
it
Thank
is
you.
possible. Yeah.
Okay. Another question from her. How injection help in the cure cancer, in curing the cancer, maybe. So, which injection, I don't know, means maybe she wants to know that is there any injections like
See,
that?
the chemotherapy is basically different types of drug we use in the cancer. There are different way to uh, give the chemotherapy. It can be given by systemic route, like via injection, or via oral route, right? So she might be asking that, yes, but it should be a continuous injecting process, like you
Hmm.
give the drips to a patient, right? When the patient is hospitalized, you give them drip. In the same way, the chemotherapy is given in the systemic route. So it is not like an injection, but it should be a continuous process of injecting that drug to the entire system. I think
Hmm.
he wanted to know about that.
Yeah, maybe.
Sorry to
Harsh,
interrupt. Can I ask one thing?
yeah, sure, Mika, please, yeah.
Just according to this question, is there any device or kind of nanoparticle mediated device which can be used in much non-invasive way to administer these drugs, like a continuous dosage?
See, there is no not non-invasive devices are not there, but invasive devices are there which are implanted. Okay, there is something called brachytherapy where radioactive
Okay.
nucleotides are just inside the device, and then the device is implanted in the body, and there continuous flow of the drug in the body. Okay, so you can just
Okay.
take it out the device as well, but I don't know any nanoparticle associated device till now. Might be there, it might be there, I might not know. So this is a good idea.
Good
Okay.
question. Yeah,
And
maybe.
another question, sorry.
Yeah, sure.
As you have told that majorly cancers are of epithelial origin, and we have studied in our BSc that epithelium is generated from the ectoderm. So, is there any way in the developmental stage that it can be marked or it can be predicted or maybe treated? Maybe it's too much, but is there any way of mapping that onset of cancer, which may exhibit in?
It is there. Few tests are there where you can test the fluid, like then you can tell that whether the kid is going to suffer from cancer or not, like whether the kid is going to suffer from any genetical disorder. Not only cancer, it can be any genetical disorder. Okay,
Okay,
it
okay.
can be told. Okay, and even
Hello.
for the patient
Uh,
also. Yeah, please.
Yeah, Kolpesh. Yeah.
okay. Yes. Sorry for interrupting, Satarupa. Yes.
No.
I have a question. Actually, I want to know from you that what is the role of artificial intelligence in screening about the cancer, screening the cancer or the hallmark of the cancer? Is there any role of artificial intelligence?
Yes, obviously, artificial intelligence it has a role. Suppose that the thing I was trying to tell now, that the story I was telling, that suppose you have 2,000 patients data with you, right? And you have the idea that which genes are going to uh, if these sets of genes are expressed, then this cancer is there. Okay, so suppose some unknown person is there, and you know their gene expression data, but you don't know whether the person is having cancer or not, right? So your system or your artificial intelligence um, associated system can tell whether the person is having or predict. It's a better way to tell. Predict. Your artificial intelligence based systems can tell whether the person is having cancer or not. That there is a role of artificial intelligence. There are many other examples as well. I'm just giving you one example.
Yeah, yeah. Actually, you were telling in your few of your first slides that personalized diagnosis. So, in our country, like not fully developed or such kind countries, how much time or how much efforts we have to give to make this personalized diagnosis treatment or the medicine for each and every people in the population of 130 billion people.
It's a very difficult question. I really don't have the answer with me as well. Uh, might be like the people in the authority, governmental authority, they can give the idea. But yes, the people who can spend money, they can obviously do it. There are few companies. I'm just sharing my screen. Um, So, suppose you want to know that whether you are having cancer or not, or if you have the susceptibility of the cancer or not, there are few companies like Map My Genome. There you can give the test. So you can. There is some things like genome patri. So you can do your blood test, and you can tell that whether you are going to have diabetes or you are going to have cancer or not. So these things are there. So if you have the ideas, like you might be knowing that Angelina Jolie, she had mastectomy because the BRCA was upregulated. Breast cancer associated gene was upregulated in her. So the thing is, if you can afford, if you can pay, you can go for this test. So you can see the diagnostic, mother and child cancer, genetic counseling, blood test. So if you go for this like DNA onco screen, and if you go for the screening, you can get to know that whether you have chances of developing any cancer or not. So it is possible. It is not that much costly as well. So just 10,000 rupees. So we are like the scientists are trying to reduce the cost as well. So these things are there.
Yeah, thank you, Madhurpati, very much for this great talk, I must say.
There are several questions in the comment section also. So one question I have online from one of my friends, Shobhik, he's asking that before differentiation of cells to perform different functions, is there any way to identify that particular gene based on epigenetic source and mute it? Is it possible?
Yes, so that there is something called single cell genomics, which is now have emerged very. It's a very new thing. So suppose the cells before differentiation, you have the cells, and for you are going with the single cell genomics, you can tell what is the genetic sequence or genetic expression of each of the cells. So that is possible.
Okay. And in the comment section, Satarupa Banerjee is asking, are electronic devices responsible for cancer?
Yes, so there are few evidence of that radiation from the mobile phones are can cause cancer. So, and there were few evidence that microwave like so. Suppose if you want to go to any like microwave is there, it's not working properly. There are chances, but the chances are minimal, and there are some limitation of that. Um, each of the company has to follow the regulation that much like electronics there should be some limitation which should be followed while repairing the electronic devices so that it doesn't cause cancer, but it might happen. Like DNA damage can happen due to the electronic device, which can eventually lead to cancer. We cannot tell, right?
Very nice.
Okay.
Just to
And
interrupt, Yeah, sorry, please, Mika. Yeah.
There's another question left. Are there evidences of using genetically modified viruses to treat cancer?
Very good
Yes,
question.
yeah, this is a very good question actually, and this is the way we do like cancer treatment. So basically, the lentiviral systems are there, which by which we can modify the genes. Like we can knock out the gene, or we can induce the gene with the genetically modified viruses, and this is how the we study the effect of the gene in the system. So but it has not come to market for the as a therapeutics perspective might be, but this is the way how we study from the molecular biology perspective, and that's why I think Choyan also got excited to got this question, right?
Yeah, exactly.
Yeah.
Exactly. Very good. Very good. Actually, Moitri, she also asked a question, but when means she's asking that lung cancer. Uh, Many patients they are recovering from lung cancer. So how is it possible? So because it's a very bad one, but still.
See, what happens is in the lung cancer, the gene which is associated is EGFR. Okay.
Okay. Yeah.
So EGFR as well, like target therapy is already there in the market, which is working very well. So that's why the lung cancer survival rate has been increased. If we know that which
Okay.
gene is causing the cancer, and if we go for the targeted therapy, the chances of the treatment options is higher, right? So
Yeah.
that's
Yeah. Thank you. Thank you so much.
I want to ask one question.
sure.
Yeah. Please.
So, ma'am, how can we increase the cancer suppressing genes?
Oh, this is also very nice question. This is the answer I have already given, but again I am giving. So cancer suppressing means that see there is always balance between the oncogene and the tumor suppressor genes, so that our body is in balance. So suppose whenever there is cancer and you want to increase, so what you can do is you can use this lentiviral systems, but it is not there in as a therapy. It is already, we know that it can be done. We can increase in the cell line, we can induce the like by via knock-in, we can induce the gene if the gene expression is down regulated. Suppose p53 is a tumor suppressor gene, and in that particular cancer the p53 is nullified anyhow, and now we want to induce p53, we have to do some knock-in study. But in the patient, knock-in is difficult. How can you can do knock-in?
Yes.
So that is the major restriction we are having now. Okay.
Okay, ma'am. Another question is after doing biopsy, the chance of the spreading the infection can be increased or triggered.
Yes, that's what I told. Na? So just now in 2022, we found that inflammation, a chronic inflammation, is a cause of cancer, right? So whenever we are just doing the biopsy, we are increasing the chances of inflammation. And whenever there is increasing chance of inflammation, there is higher chances of metastasis.
Okay, ma'am. Okay.
Okay.
And how can
Yes.
we detect the, I mean, differentiate between cancer cell and the normal cell after staining?
So there are different ways. Okay, you can like if you do histopathology, normal H&E staining. So in the cancer cells, um cytoplasmic the cytoplasm will be less, and your nucleus will be bigger. So nucleus to cytoplasmic ratio will be high in the cancer cells than the normal cells. The cells become shrink. Okay, this is called NC ratio. So NC ratio is increased, and the cells dysplasia is increased. There is hyperplasia. So there will be more number of the dysplastic cells. You can find that a lot of immune cells are there. So there are different a few features by which you can tell that which are cancerous cells and which are normal cells.
Okay, ma'am. Thank you. Thank you, ma'am.
This is a part of pathology
Um,
actually.
I
Yeah.
I have another question from Tithi. Actually, I missed that question. Why should we care about tumor microenvironment?
This is a very nice question again. So, tumor microenvironment is basically a place where, like, to a tumor to develop, you need specific things. First thing is you need blood supply, so that you get the cells get food. So to give the blood supply, their new blood vessel is generated. This is process is called neoangiogenesis. Okay. Now, when new blood vessel is there, with the new blood vessel, there are new inflammatory cells as well, like cancer associated fibroblasts are there, then some other cells are there which support the cells to survive. Okay, this cancer associated fibroblast and uh, like triad of this macrophages, cancer associated fibroblasts, if it is there more, it basically is helping the tumor cells to survive. So this is given the survival, the tumor microenvironment is given the cells survival advantage. So this is why the tumor microenvironment is very, very important. And there is expression of different types of growth factors, like vascular endothelial growth factors, um, which basically support the tumor cells to grow. So that's why this is very important.
Thank you so much. Any other questions? Feel free to ask.
I really enjoyed the session because there were lots of questions, and the questions were quite interesting as well.
Yeah.
Another question I have.
Undoubtedly very interesting talk. Very interesting talk. So, yeah, any other questions, please?
Yes, Chand, I have.
Yeah, please. Yeah, sure.
So, ma'am, you have there was a slide where you showed that 12 signs about cancer, 12 warning sign of cancer. So, abnormal menstrual cycle. What is the reason behind this, Mane?
So, see, if there is ovarian cancer or if there is cervical cancer, so you will find spotting as one of the major issue. And see, like the hormonal, even in the breast cancer, there is hormonal alteration. So, whenever there is hormonal alteration, obviously the menstrual cycle will be changed, right? So, that is the reason.
Okay.
So many cancers are there, like which are hormone associated: breast cancer, cervical cancer, ovarian cancer, right?
Okay, ma'am. Okay. Thank you.
Hello,
More
ma'am.
questions? Yeah, Tanmoy, please.
How can knowing the mythos and death cause a patent cancer safe treatment?
Can you please repeat your question?
How can knowing the mythos and death cause a patent cancer safe treatment?
তুমি একটু বাংলায় বলবে ভাই, আমি বুঝতে পারছি না কোশ্চেনটা।
বা যদিও লিখে দেয়,
হ্যাঁ, লিখে দিলেও হবে।
Tanma, can you write the question in the comment section?
Yeah, he's writing maybe.
Your audio is breaking.
না, কিন্তু খুব ভালো হলো আজকে, একদম এরকম কোয়েশ্চেন অ্যান্সার হবে তবেই তো একটা টক ভালো লাগে আর কি।
Yeah, I really enjoyed.
এবং অনেকেই জয়েন করেছিল, ভালো লাগলো। এবার এটা যেতে গেলে তো আরো ভালো হবে আর কি।
Any other topic?
Meanwhile, can I ask a question?
Yeah,
Yeah,
sure.
please.
I don't know, but what is the condition, or what was the condition in COVID in case of cancer patients who are already suffering from cancer?
It is a very interesting question. So the thing is, during cancer, what happened when the people who were suffering from COVID, the COVID, like since they were immunocompromised, so what we knew that the COVID was like for 14 days, the COVID was supposed to be the burden of the virus load should be reduced. But what it was found that for a patient, it was more than 120 days the burden was there. So the thing is, the same thing happened with, like, in my personal experience, I can also tell, because my father was suffering from cancer, and it was diagnosed in last November. And when his chemo was started, he was also diagnosed with COVID. And it was very unfortunate that after 14 days, when it was tested, there was no change. So yes, in the immunocompromised patients, if the COVID was there, it was it is really a difficult situation, because the viral load was there for a long time. It is not only I'm telling from the textbook or news, I'm telling from my own experience as well.
Okay.
Okay.
It was dangerous at some point.
And the chemo had to be stopped. It was very unfortunate that after 10 days of first cycle, the chemo has to be stopped. It was I might be being emotional, but it was the scenario.
Yeah.
This
Okay,
is a very interesting question. Yeah.
we have a question here. Yeah, can PC? Okay.
Yes. So the thing is, there are high chances of cancer. Like, not due to PCOS. This is a very controversial topic. Whether PCOS can lead to cancer. So the PCOS is associated with other diseases like metabolic syndrome, and the if the person is having PCOS, their chances of taking oral contraceptive pill to regularize the menstrual cycle is there. And when you are taking OCT for a long time, obviously there can be chances of, like, it will give you support for the ovarian cancer, but will be increased chances of breast cancer. So yes, that can. There is some correlation, but much more studies are needed. So one of my student is here, Uma is there, who have joined in the talk. So she was very interested to do her research in this topic, but since we are not having much sample, so we could not start the research. So okay, how can knowing the mutation that cause a patient cancer shape treatment? So this is the point of my entire talk, right? So mutation is one of the genetic alteration, and since we know about the mutation, then only we can go for the targeted therapy. So obviously, knowing about the mutation that cause a patient's cancer, obviously helps in the deciding the treatment strategies.
Very nice. Very nice. Any more questions? Maybe Srinanti wants to know that. Okay. I didn't understand her question. Okay. Well, yeah, for the injection thing, you already answered about the injection and other things. So he's asking that what are the components of the injection maybe.
Hello.
So yeah, no, please.
Hello, ma'am. Hello, ma'am. I have a small question from you. Recently, I came across through some paper and came to know that steroid medicine, if somebody is taken in a like constant manner, there can be a reduced chance of getting a cancer. Like I came to know from one of the person, professor, basically in a class. So I just want to I just want you to highlight on this. Like it can be useful. So how like steroid consumption can like reduce the risk of cancer actually?
So what is steroids? Steroids are mostly, like, they are used for the treatment of the mostly immunological consequences. Okay, so they are basically given to the immunocompromised people. Okay, for increasing the immunity. So basically, one thing is, as I have told, one of the major hallmark is um, immunity, right? Like. So the thing is, whenever you are boosting immunity using steroid, okay, like basically, so that is the way that might be the case where using steroid might decrease the rate of causing the cancer. I really don't know much about the topic because you are telling that you have studied in some research paper. So might be these are the new things which are coming to the picture. But I suppose it should be the case, if I'm not
Yeah,
very wrong.
and I can remember there are some issues of steroid-induced cancer also. When I was in my master's, I was reading some articles like that. So steroid overdose of steroid at some point can have some kind of like in COVID also we saw that overdose of steroid had different side effects on the immune system
Yeah,
also.
it might happen. I need to study more about this topic.
Yes, sir, that's true. Like steroid, if taken in a high amount, like that is not favorable for the body. But few suggest that if you are taking in a very less amount, in a prescribed amount, then basically it can reduce the risk of cancer.
So
And this,
another thing is that non-steroidal inflammatory drugs are also used for reducing the cancer. Yeah,
yeah, yeah, yes, ma'am.
you have to tell.
Hi Satarupa, it's a good. Your talk is very good and very nice. Can I add something about this steroid thing?
Sure, sure. I will be happy to know.
I think that actually steroid use for the reduce the inflammation. When non-steroidal anti-inflammatory drug is not working, in that condition, steroid apply. Doctor prescribe for the steroid. So in that way, that it reduce the inflammation, so it can reduce in the risk of cancer. I think so. I just by concept. I haven't read,
That is the
but
that is the thing I also told. I also told the same thing. That might be that is the thing. I'm not very much sure. So might be that is the
I am
thing.
not
I
sure,
agree
but
with you.
I think in that way that is the root of that can be the cause for that reduce the inflammation, and inflammation and cancer is highly linked. So you
That is the
also
thing I told
told
her
in
that
this.
22 that inflammation is a new hallmark. They have been added.
Mm-hmm.
So I also so I agree with you.
Thank you
Okay,
so much,
thank
ma'am,
you.
for this. Thank you.
Thank you.
So we have another question.
Hypothyroid. Yeah. So as I was telling, that hypothyroidism is also associated with PCOD and metabolic syndrome. So all are associated, and like there are not much evidence that hypothyroidism can lead to cancer, but like that also have to be taken. Like I have to check much more on that topic, like the association of hypothyroidism and cancer.
Can I add something?
Sure.
Actually, in hypothyroidism, metabolic rate is reduced, so you can gain the weight, and obesity will increase. So, and obesity is highly, all are indirectly related, but obesity is highly linked to cardiovascular cancer and diabetic thing. So in that way, can indirectly that it reduce the metabolic rate, so you can gain weight, and obesity is highly linked to cancer. So in indirect
That's a
way
good
that can.
correlation.
Currently, I am working in obesity. That's why I think that is the root.
Okay,
Okay,
probably the last
carry on.
question is coming. Srinanti wanted to ask, actually, she clarified her question that what are the components of cancer vaccine or vaccines against cancer? She wanted to know.
See, different cancer vaccines have different mechanisms. So basically, the cancer vaccines basically used to give you the memory against the, like for the cervical cancer might be. So cervical cancer is HPV associated, that human papilloma virus associated. So the HPV vaccine is or the hepatitis B virus vaccine. So they give the memory against the uh, genomic alteration that produce different types of antigen. So this is the way these are the components, or this is the mechanism by which the cancer vaccine works. I hope you got your answer. But if I go into detail, it will be little bigger. So basically, it gives you the memory against the antigen which causes the host cancer due to the viruses.
Yeah, actually, at the end, everything is very interrelated. What Moushamidi is saying, actually, yeah, very interrelated with cardiovascular thing, and because when I was writing this ferroptosis thing, and so many things, because I was mainly concentrating on neurological challenges, but yeah, everything was there, from cardiovascular to diabetes, obesity, everything was linked. So I had to choose only the neurological things from there. So it's a very very interesting thing. Maybe when we do these crosstalk things, ultimately at the end of the story, it becomes very complicated, because you can link all of the pathways at some point, and it becomes very complex. So yeah, everything is linked. Yeah, it's very very nice actually.
And in obese condition, a certain amount of the chronic inflammatory condition is continues.
Increased.
And there is some common pathways which are activated in cancer and obesity. So that's why they are linked.
Absolutely.
Okay. One thing, so sorry, it's not related, but one thing I could remember, when I was doing my master's, so I was in Shelidhi's lab, and I was working in Shelidhi's and Shamida's lab both. So there was a news from Jadavpur University that they had one researcher or a research group. They actually applied for a patent for this rheumatoid arthritis and other things. They made one ointment, something like that. And later, Samita was telling, and it was a big issue at some point, because that particular ointment, it was having overdose of steroid, which was not at all prescribed. What we say as prescribed, it was more than the
within the
limit.
prescribed limit. It was not within the prescribed limit.
Yeah, exactly. And it was causing permanent damage due to the inflammation. So that same thing is coming again. So
basically, yes, all are related, like hypothyroidism, PCOS, obesity, cardiovascular disease, cancer. So basically, all are related. So
and the cell death also. So if you will go to the cell death thing, so it will be like a huge tree again.
Absolutely.
Any other questions? I know Satarupa is very hungry, because it's now maybe 10 o'clock there, over there.
No, it is 9:30. But it was very interesting session, and I felt like I felt good answering those questions, and people are interested in the talk. So that's why they are asking questions. So I'm really happy for that. And another question I'm seeing, that why are older people more prone to cancer in general? This thing I have already answered during my talk, because the DNA damage repair system can't solve all the problems, and they keeps on accumulated, and the older people have higher chances to get cancer, because the mutations are not cannot be fixed by the DNA damage repair systems. So
very nice. Thank you, Satarupati. So I don't think there are many more questions, but yeah.
Thanks, Choyon, for inviting me, and it was very interesting, nice session. I really enjoyed, and you can call Moshumidi also sometime in this group. So
yeah, we are very happy that Moshumidi could join us, and if at some point she will be free, I will request her to give a separate talk on her research topic if she wants. We will be very happy.
Yes, Choyan, you are always welcome, and I'll be happy to join this.
Thank you so much.
And every time in every week, I and Satarupa generally talk about various things, personal thing and research thing, and always we are contact with her.
Very nice, very nice, very nice.
And sometimes I take suggestion and guidance from Satarupa also. She is my senior actually.
Ah.
Thank you.
Before to
Thank
end
you,
the
Satarupa.
session, sorry. Yeah.
Thank you, Satarupa, and for your nice talk and inviting me for joining.
Yeah, it was very nice to have you as well, and your ideas also helped us. Your opinion also helped us.
Exactly. Yeah, so before to end the session completely, actually from my side, I will just thank Mihika and Kolpesh. Actually, Mihika is the host of the meeting, and Kolpesh did the poster.
It was very nice
For you.
flyer. It was very nice flyer, and thanks everybody, the entire team.
Yeah, and yeah, everyone
Thank you.
was so excited.
Thank you, Shatrapati.
And Shourish is also there, but Shourish actually could not join. He actually take care of the emailing and other things of Journal Club, officially appointed from Ethophilia. But yeah, he's not here. So thanks to Kolpesh, and Kolpesh was also taking care of this technical thing, so he's actually recording. So thanks to Kolpesh and Mika. So Mika will finally conclude just a Thanksgiving for 2 or 3 sentences, but before he starts, I will just say, Satrupati, we will be very happy to collaborate. Yeah, we are not very that professional to collaborate, but yeah, if you think that okay,
I
we
will be
can do
happy
some
to.
work,
I will be
or
happy to join.
we can write a paper, something like that. So we will be very obliged. So thank you very much.
Thank you very much.
Mika, please.
So we are more than obliged that you joined and gave this wonderful talk today, and you made this complex topic so much lucid to us. So it's a big thanks to you, and definitely we wish to meet you offline also. So
I will be happy.
and listen to your talk in offline. That would definitely be a joyous thing to listen. So